Hello everybody, my name is Dr Owen Kavanagh and I'm a senior lecturer in Biomedical Sciences at York St John University. One of my research and teaching interests involves the study of viruses and how we protect ourselves against viruses, basically our immune system. If you caught my previous video, we basically explained what is a virus using coronavirus as an example. And today I thought it was appropriate to ask the question, how will we defeat SARS-CoV-2? If you remember, SARS-CoV-2 is the name of the virus, and COVID-19 is the name of the disease caused by the virus. So I believe with the possibility of global eradication of this disease, we have one principal way of doing it, and that's using our own immune systems and basically vaccinations. So in this small video, I'm going to explain the principle behind vaccinations and also why we need to err on the side of caution and take 12 to 18 months to develop a vaccine. Great, so to talk about the immune system, it's quite a complex system, but basically there's two arms to it. One is called the innate immunity, which consists of some cells, our skin, teardrops, sweat, inflammation. So these are non-specific ways that we can prevent a pathogen from entering our bodies. This is good because it's short-term, fast-acting, but once the pathogen gets beyond this defense system, then the person is vulnerable to infection. This is where the adaptive immunity comes in, and it'll be the focus of my talk today. So the analogy I always use for adaptive immunity is they're attack dogs. So we're able to train these dogs to sniff out a particular pathogen, and they will hunt around the body, and when they find that pathogen, they will jump on it and viciously attack it. So that's what these cells are. And the actual cells are called lymphocytes. So I'm presuming you've probably heard of this term, lymphocytes. That's what they are. So lymphocytes come in two forms. There's the B cells, and that's these angry guys, and they are produced in the bone marrow and they mature in the bone marrow, hence why they're called B cells. And on the other side, we have the T cells, and that's these guys. These guys also produce in the bone marrow, but they mature in the thymus, hence why they're called T cells. So these are the two sets of lymphocytes we're going to focus on for this video. So B cells, you might know this, but B cells actually produce this antibody that everybody's on about. So antibody is really important. It's really easy to measure. So that's why all the scientists are looking to see, will our antibodies protect us? You might have seen the plasma experiments. That's basically transferring antibodies from an infected but cured person into someone who's not infected, and there has been evidence to show that antibodies can protect against this virus. So the B cells produce lots and lots of this antibody, and basically what an antibody is a protein molecule. It's Y-shaped, and at the two tips of the Y shape, they're parts that can recognize specifically the virus in question. It can be bacteria as well, but in this case, the virus. So by this I mean it will recognize, in particular, the spike protein on the coronavirus. It will recognize the influenza spike protein. It's something different. It's very specific, and that's why we use the sniffer dog analogy. It's able to sniff out coronavirus and attack it viciously. So what it does in this case is it kills the virus, or as we call it, neutralizes the virus. So just a little recap to show how antibodies work. There we have our virus. We have the genome in the middle, surrounded by the lipid bilayer, the blue lipid bilayer, and then the yellow spike proteins protruding from it. So in order to gain entry to a cell, to infect a cell, we need to have a receptor. So the receptor is the lock, and the S or spike protein is the key. So once the key fits into the lock, the virus is able to infect the cell and produce lots and lots of different progeny, which go on and infect neighboring cells, but also transmit the virus to other people. So in the case of antibodies, what they do is they bind to the S protein and they prevent the cell entry. And what that means is you don't get infection. As you can see, the virus tries to enter the cell, but without being able to bind to its lock, it can do nothing. The T cells have a slightly different approach. When a virus cell infected cell, it expresses the parts of the virus on the cell surface as a sort of help signal, as an emergency red flag. These T cells are able to sniff that out. There's different types of T cells, but the ones we're going to focus on are called killer T cells. I think the clue is in the name. So these killer T cells come in and they recognize this foreign protein, this invader on the cell surface, and what they do is they savagely kill the cell, so the infected cell is gone, and then the rest of the immune system can come in and sweep up the damage. The key thing that differentiates these cells from other immune cells are they actually have memory. So B cells and T cells, once they encounter the virus, they're able to produce many copies of B cells that will produce the same antibodies against the virus. But what these do, and along with the T cells, is they will patrol the body for possibly years, just waiting to sniff out the reoccurrence or reinfection of this pathogen, and then they will explode into action and savagely kill the virus in question. So just to look at the time scale of this, if we look on the left-hand side, we have the strength of the immune response. And on the bottom axis, we have the time in months. This will be post-infection. So we have an infection on day 0. What happens is we launch a small, weak B and T cell response. It's not that great. But the key to it is if we have a secondary infection, we launch a massive immune response. This is because of the memory created in the first stage, and also those B and T cells, they keep on training themselves to be better and better killers of the coronavirus. So this is where we're at now. The question is, people who've been infected the first time, will they be protected from the infection a second time? The signs are looking good at this, but it's still early days. This is also the principle of vaccination. So you give an initial vaccination, which mimics the primary infection, and then you give a booster infection to generate this massive response. And remember, those memory cells will be there patrolling the body, hopefully for many years afterwards. So a final note of caution on this. The immune system is a double-edged sword. So on one hand, hopefully, it causes disease protection. So vaccination can protect us against the disease. But we have to be careful that the vaccine doesn't stimulate an inappropriate immune response. You might have heard of these cytokine storms. Part of the whole disease with this coronavirus is it actually drives an inappropriate immune response. So we have to be very careful with this double-edged sword. That's why we have to do a lot of clinical trials to work out the safety and efficacy of this vaccine, because there's a lot of people out there probably waiting for this not to work so they can validate their opinions on vaccines. That's why it's going to take 12 to 18 months, and it is necessary because the whole world is watching. So just a final note, the testing everyone's talking about. So the swabs they're doing, that's actually looking at the viral genome. Okay, so that tests for a current infection. If you have the viral genome, you're most likely to have the virus inside your body. The other test they're talking about is where they're going to measure the antibodies, and that will measure previous infections, and that will allow epidemiologists to track how many people actually have been infected, because I can guarantee you only a small subset of the people that turn up in hospitals and are used for the statistics are actual COVID positive patients. That's great. That's the end of this. So if you've any questions to ask me, please drop me a line. We have a Facebook page and we have a Twitter page. I'd also like to thank at this point Dr. Michael King, who helped me with some of these animations. Thank you for your attention, and I hope to hear some questions from you soon.